Good afternoon. May I request all participants who are joining through the WebEx to kindly mute their microphone. Kindly mute your microphone, please. Yes, thank you.
Good afternoon, everybody. So we are now starting the post-lunch session. So in this session we have 4 speakers. So the first speaker is the keynote speaker, and let me briefly introduce the first speaker of this session, Professor Hiroyuki Ushida, and let me briefly tell his biographical sketch. Professor Yoshida, he is the Associate Professor of Radiologist, Harvard Medical School. He is also Director of 3D Imaging Research, Massachusetts General Hospital. From January 2005 to presently, he is working at Harvard Medical School for more than 15 years in the area of early diagnosis of colorectal lesion with CT colonography, radiomic decision support system, machine learning for colon cancer prevention, and CT-based biomarker for predicting clinical outcome. So he is the speaker, he is the keynote speaker for today's afternoon, and I would like to invite Professor Yoshida for his post-lunch keynote address. Thank you.
At the Massachusetts General Hospital and Harvard Medical School in Boston, United States. In this presentation, we will talk about AI imaging biomarkers and how to use them for prognostic prediction. We will discuss 2 example applications, interstitial lung diseases, and the COVID-19. So, what are AI imaging biomarkers? In radiology, we usually obtain some sets of clinically acquired images from a patient, such as a CT image dataset. The basic idea of the AI imaging biomarkers is to use artificial intelligence, or AI, to automatically extract useful quantitative features from these clinically acquired images. These features can then be subjected to a survival model to obtain a biomarker, which can be used for making predictions about the disease progression or prognosis of the patient. Such an AI imaging biomarker can be very useful as a clinical decision aid for making informed decisions about the treatment options and therapeutic management of the patient. We have recently developed two kinds of AI imaging biomarkers based on deep learning. The first biomarker, which we call radiomics, is a supervised method where we train a convolutional neural network, or CNN, to extract useful features from the images for performing an explicit survival analysis for the prognostic prediction. The second biomarker is called GAN radiomics, and it is a weakly unsupervised method where we use a generative adversarial network, or GAN, to come up with the prognostic prediction directly from the images. We will discuss both of these methods in a moment. For these AI biomarkers, we will consider two example applications. Initially, when our team started developing these AI imaging biomarkers, we focused on the prognostic prediction of patients with interstitial lung diseases, such as the idiopathic pulmonary fibrosis, or IPF. More recently, we have also applied these biomarkers to the prognostic prediction of patients with the COVID-19 pneumonia. So why use AI imaging biomarkers in these diseases? Let's use the IPF as an example. The IPF, or idiopathic pulmonary fibrosis, is a major chronic progressive interstitial lung disease. It turns out that the median survival time of a patient with IPF is only about 2 to 5 years from the time of the diagnosis, and this is obviously poor. Another problem is that the disease progression of IPF patients is highly variable. Therefore, it is difficult to make accurate predictions about the progression and mortality of patients with IPF. Because of the clinical importance of IPF, several prognostic biomarkers have been proposed for the prediction of the disease progression and mortality of IPF, and some of these biomarkers are shown here. In particular, GAP index, which is a physiological biomarker derived from pulmonary function test, is currently considered as the most established prognostic biomarker for patients with IPF. However, there are no established imaging-based biomarkers for IPF. Although some radiological features based on chest CT images have been identified, these are mostly based on visual assessment by radiologists. Therefore, it would be very useful if AI could be used to provide objective and reproducible predictions about the IPF progression and mortality based on the CT images of the patients. We will next take a look at the two AI imaging biomarkers we have developed, including some of the results that we have obtained with the IPF and COVID-19. First, we will discuss our supervised AI imaging biomarker, called radiomics. The radiomics biomarker combines a supervised net convolution neural network with a traditional survival analysis methodology to perform survival predictions. Here is a schematic overview of the radiomics scheme. The deep learning net component is shown on the left, and the survival analysis component is shown on the right. The net is a well-known fully convolutional neural network that has a contracting section and an expansive section. These two sections are separated by a so-called bottleneck layer. There are also skip connections between the contracting and expansive sections. It turns out that such a design is very effective for performing semantic image segmentation tasks. As a first step, we train the net to segment chest CT images into clinically meaningful tissue patterns. To collect training data, an experienced pulmonologist manually extracted and annotated over 4,000 regions of interest on chest CT images of patients with interstitial lung diseases. The regions were classified into 5 lung tissue patterns, including ground glass opacity, honeycombing, reticulation, consolidation, and normal tissue pattern. The image on bottom right shows an example of such an annotated image. Here, more than 5 tissue categories are shown, but the extra categories are combinations of the basic 5 patterns. The labeled axial CT images are used to train the net for the semantic segmentation of the CT images into the 5 lung tissue patterns. After the training, we can apply the net to perform a semantic segmentation of the chest CT images of new, unseen patients, who were not part of the training dataset. Such a segmentation works well, but for the purposes of the U-radiomics model, the point is that, after the training of the U-net, the bottleneck layer of the U-net, or the feature map between the contracting and expansive sections, contains critical information about the interpretation of chest CT images. Therefore, we can use the output of this bottleneck layer as a feature vector for our survival analysis. The features of the bottleneck layer basically tell us everything we need to know about the input image. We call these features as the U-radiomics feature vector. Once U-radiomics vectors are obtained from all the CT images of the patient, the median of these vectors is identified as the U-radiomics vector for the patient. In the second part of the U-radiomics model, the feature vector from the bottleneck layer of the U-net is subjected to a survival analysis. For the survival analysis, we use the elastic net Cox model, also known as the Cox proportional hazard model with elastic net penalty. As the name suggests, this model maximizes the likelihood shown in the middle of the slide. The first part of this likelihood is the usual partial maximum likelihood estimation of the Cox hazard ratio. The second part is an elastic net penalty, which consists of L1 and L2 penalties, with a blending parameter alpha. Because of this formulation of the objective function, the model can perform simultaneous feature selection and optimal estimation of the hazard ratio. The output of the elastic net Cox model is compared with the true survival time of the patient. Now, we go through some of our results about the prognostic performance of the U-radiomics when it is applied to interstitial lung diseases. As a test patient cohort, we retrospectively collected 75 patients with idiopathic pulmonary fibrosis, or IPF, from our institution. The cases had been collected between 2005 and 2015, and they were completely independent from the cases that we used to train the U-net of the U-radiomics model. All these patients underwent thin-section CT scans of the lungs, as well as standard pulmonary function tests, as shown at the bottom, to obtain the physiological CPI and gap index values. The gap index and CPI are prognostic biomarkers that predict the mortality of patients with IPF. They can be calculated from pulmonary function test results, in the manner shown on this slide. This slide shows the basic statistics of the demographics, pulmonary function test results, and the distributions of the CPI and gap index values of the test patient cohort. For these patients, the CT scans were performed with full inspiration and in supine position, from the lung apiece to the bases, by using the CT scanning parameters as shown on this slide. For the metrics for the prediction performance, we used the concordance index, or C-index, which measures the concordance of actual and predicted survival. The value of C-index ranges from 50% to 100%, where a higher value shows a better performance. We also used a patient-based bootstrapping with 500 replicates to obtain an unbiased estimate of the C-index values and their confidence intervals. We then compared the predictive performance of the U-radiomics with those of the CPI and the gap index. We also evaluated the risk stratification performance by dividing the test patient cohort into low and high risk groups based on the mean of the predicted hazards, where the group separation was tested by use of the log rank test. Here is the result of the comparative performance. The U-radiomics had a higher C-index value than those of the CPI and the gap index, and this improvement was statistically significant. We also tested the performance of the combination of the CPI, gap index, and U-radiomics, but this did not improve the performance. These results suggest that the U-radiomics provides a much more comprehensive assessment of the IPF than do either of the CPI or the gap index. Here is an example of the survival estimation for a single individual from the test patient cohort. This is an 82-year-old male patient. The survival curve estimated by the U-radiomics looks like this. The estimated median survival time, which is shown by the dotted lines, was 442 days, and this is very close to the actual survival time of 437 days of the patient. On the other hand, the survival curve estimated by the gap index looks like this. The gap index predicted a much longer median survival time of 1712 days, and the survival curve is much more spread out. The situation is the same for the CPI, as shown here. This example demonstrates the superiority of the radiomics in estimating the survival curves of individual patients. This slide shows the Kaplan-Meier survival curves for the low and high risk groups of patients stratified by the radiomics, as shown on the left, and by the CPI, as shown on the right. The difference between the low and high risk groups stratified by the radiomics was statistically significant, whereas the difference was not significant for the CPI. On the other hand, in the case of the gap index, as shown on the right, the separation between the low and high risk groups was statistically significant. However, we can see that the radiomics separates the groups much better than does the gap index, with virtually no overlap between the confidence intervals. These results demonstrate the superiority of the radiomics in risk stratification as compared to the CPI and the gap index. To summarize, radiomics can be used to perform image-based prediction of the survival of patients. Our results show that the radiomics outperforms the existing physiological biomarkers in predicting the overall survival and risk stratification of patients with idiopathic pulmonary fibrosis. This indicates that the radiomics has the potential to be a highly effective image-based prognostic biomarker. Next, we will introduce our second AI imaging biomarker. This is a weakly unsupervised GAN radiomics model, which we also call as PIX2-SERV. As the name suggests, the model is based on a generative adversarial network. So, in the previous section, we saw that the radiomics model already works quite well. Why would we need a second prediction model? Well, there are several reasons. One of the problems of the supervised learning approach of radiomics is that we need to annotate a training dataset for the net. Manual annotation is very laborious, costly and time-consuming. You also need to keep annotating new images if you collect more training cases or if the imaging technology improves and provides better images that replace the existing ones. Another problem with the supervised approach is that the performance can depend on the accuracy of the annotations. Any errors in the annotations can propagate into the prognostic prediction. There are also cases where you just cannot express the ground truth simply as a precise binary decision for the annotation. This is why we have also investigated the use of weakly unsupervised learning, or GAN radiomics. In this approach, no annotations are needed, other than the survival time of the patient. The GAN radiomics simply takes a look at the images and comes up with a survival prediction. In the next slides, we will take a look at the GAN radiomics model in more detail. A recently reported paper showed that a conditional GAN could generate better predictions than traditional survival analysis methods, such as the Cox proportional hazard and accelerated failure time. The approach also had the advantage that the distribution of the survival time can be estimated directly from the input predictors. However, this conditional GAN approach was not based on images but epidemiological data. Therefore, our approach was to develop a conditional GAN model, called PIX2-SERV, which can estimate the distribution of the survival time directly from chest CT images. This slide shows the overall architecture of the PIX2-SERV model. The model is a conditional GAN that has a generator network called the time generator, and a discriminator network called the discriminator. The input to the PIX2-SERV model are the original input CT images. There is no need for making annotation of lesions. The time generator takes the input CT image and calculates the corresponding estimated survival time image. The time generator has an encoding block, which consists of 4 convolutional layers as shown on the left. On the right, we see that the output of the encoding block is vectorized and forwarded to a prediction block that consists of 3 fully connected layers that calculate a survival time value image. Actually, the original output of the convolution network is a single survival time, but we blow that up into a survival time image that has same dimensions as the input image, and every pixel has the same time value. The second part of the PIX2-SERV model is a discriminator network. The input to the discriminator consists of the original CT image and corresponding survival time image. There are 2 cases, but the CT image is the same in both cases. The survival time image can be either the one estimated by the time generator, a so-called estimated survival time image, or it could be based on the real survival time of the patient, and here it is called the observed survival time image. The output of the discriminator indicates if the input pair of the CT image and the corresponding survival time image looks real or fake. If the discriminator says that the output looks fake, it thinks that the survival time image was generated by the time generator. On the other hand, if the discriminator says that the output looks real, it thinks that the input survival time image is based on the real observed survival time. As usual, the Pix2-Serv GAN is trained by training the time generator and the discriminator in a feedback loop, where both networks improve their performance over time by trying to fool each other. The loss function consists of the usual adversarial loss of a conditional GAN shown by red underline, but there is also a survival loss term shown by blue underline. The survival loss is unique to the Pix2-Serv model. For non-censored patients, the survival loss is the same as the L1 loss between the observed time and estimated time values. However, for censored patients, where the death was not confirmed at the observed time, the survival loss penalizes the time generator only if the estimated survival time is earlier than the observed survival time. After the Pix2-Serv model has been trained, we use the time generator to estimate the survival time for new previously unseen patients. As with radiomics, we first estimate the survival times from each axial CT image of the patient, and then we use the median of these survival times as the estimated survival time for the patient. Now let's take a look at some of the results we have obtained with this GAN radiomics. The test patient cohort was the same as that we described previously for the evaluation of the radiomics for idiopathic pulmonary fibrosis. Similarly to the radiomics, we compare the predictive performance of Pix2-Serv with those of the CPI and the gap index. Again, we used the concordance index as the metric of the prediction performance. We also calculated the so-called relative absolute error, as defined here. To obtain an unbiased performance estimate of these metrics with confidence intervals, we performed the evaluation by use of bootstrapping with 50 replicates. Here is the result for the concordance index. We see that the Pix2-Serv yielded much higher performance than those of the CPI and the gap index, and that the improvement was statistically significant. Here is the result for the relative absolute error. We can see that the Pix2-Serv had much lower error rate than those of the CPI and the gap index, and this was again statistically significant. Here are the Kaplan-Meier survival curves that were stratified into low-risk and high-risk groups by the median estimated survival time obtained from the Pix2-Serv, the gap index, and the CPI. The log-rank test showed that all these models differentiated the low and high-risk group with a statistically significant separation. However, with Pix2-Serv, we can see that the separation was clearly largest, and that the confidence intervals of Pix2-Serv do not even overlap. This slide shows the survival curves for the IPF patient cohort, as estimated by Pix2-Serv, the gap index, and the CPI. The overlapped blue curve shows the actual survival time curve. Visual assessment of these curves indicates that the survival curves estimated by Pix2-Serv approximate the actual survival times much better than do those of the gap index and the CPI. This slide shows the estimated survival time of a 51-year-old male patient as boxplots, as estimated by the Pix2-Serv, the gap index, and the CPI by use of the patient-based bootstrapping. The actual survival time was 257 days, and it is indicated by the red dotted line. We can see that the median of the survival time estimated by the Pix2-Serv model is more accurate than those of the gap index and the CPI. To summarize, the GAN radiomics model or Pix2-Serv is based on a conditional GAN, and it can be used to estimate the distribution of the survival time from the chest CT images of a patient. In our experiments, the Pix2-Serv has outperformed physiological indices, such as the CPI and the gap index, in predicting the overall survival of patients with idiopathic pulmonary fibrosis. This indicates that the Pix2-Serv model can act as an effective prognostic biomarker for image-based prediction of the overall survival of patients. In this final section of the talk, we will take a look at the preliminary results that we have obtained with the coronavirus disease 2019, or COVID-19. As you know, more than 686,000 people around the world have died because of the COVID-19 since this past January. Because of the rapid spread of COVID-19, fast, accurate, and early clinical assessment of its progression and mortality is vital to support clinical decision-making and logistic planning of related healthcare resources such as hospitalization, ventilation, and ICU admission. However, similar to what we observed previously with the interstitial lung diseases, the clinical course of patients with COVID-19 is highly variable, ranging from asymptomatic to severe respiratory failure. Therefore, the prediction of the COVID-19 progression and mortality is challenging. Several prognostic biomarkers have been suggested for COVID-19 in the literature, such as patient demographics and laboratory tests as shown on this slide. Another possibility would be to use imaging methods, such as high-resolution chest CT, as prognostic biomarkers. In chest CT in particular, the lung tissue patterns are highly correlated to COVID-19. In its early phase, COVID-19 is indicated by ground glass opacities. In later phases, it is indicated by consolidation and crazy paving patterns. Indeed, several quantitative variables based on chest CT images have been reported to be associated with COVID-19. This slide shows some examples. However, there are no established imaging-based prognostic biomarkers for COVID-19. We already showed that our two AI imaging biomarkers work very well on chest CT images for idiopathic pulmonary fibrosis. Therefore, we have also applied these biomarkers to the prediction of the survival of patients with COVID-19. In the following, we compare the performance of the AI imaging biomarkers with those of blood tests and the image-based biomarker of the extent of aerated lung parenchyma, or volume wall, shown below. The low values of the volume wall are associated with a poor diagnosis. For our experiments, the U-radiomics and GAN-radiomics models had been pre-trained as we described previously. As a test patient cohort for the COVID-19 experiments, we retrospectively collected 214 patients who had been diagnosed as COVID-19 positive using the reverse transcription polymerase chain reaction technique. All of these patients had the thin section CT scans of the lungs and blood test results available. Here are some basic statistics of the demographics, blood test results, and CT scan parameters for the COVID-19 patient cohort. We evaluated the prognostic performance of the biomarkers on this patient cohort in the same manner as we did with the idiopathic pulmonary fibrosis. Here are the results of the prognostic performance of U-radiomics in comparison to the volume wall and the blood tests. We can see that the U-radiomics produced a very high concordance index value of 87%, which was over 24% higher than those of the blood tests or the volume wall. These differences were statistically significant. Also, the GAN-radiomics or PIX2-SERV model showed a high C-index value of 84.4%. The C-index value of PIX2-SERV was over 19% higher than those of the volume wall and the blood tests, and these differences were statistically significant. This is the result for the relative absolute error with PIX2-SERV. We can see that PIX2-SERV had a statistically significantly lower relative absolute error than the other predictors. Here are the Kaplan-Meier survival curves of the COVID-19 patients, stratified into low-risk and high-risk groups by the median of the estimated survival times. The volume wall did not produce a meaningful separation, whereas both PIX2-SERV and the blood tests produced a statistically significant difference between the patient groups. We can see that the separation between the patient groups was largest with PIX2-SERV. Here are the estimated survival curves of the COVID-19 patient cohort. The actual or true survival curve is shown overlapped in light blue color. Visual assessment of the survival curves indicates that the curve estimated by PIX2-SERV approximates the actual survival times better than the blood tests, and much better than the volume wall. Finally, this slide shows a comparison of the estimated survival times for a 60-year-old male patient. The survival times were estimated by use of patient-based bootstrapping. In the box plot on the right, the red dotted line shows the actual survival time, which was 14 days. We can see that the median survival time estimated by the PIX2-SERV is more accurate than those of the blood tests and the volume wall. In conclusion, we have developed AI imaging biomarkers that use AI to automatically extract useful image-based features for making prognostic predictions about the disease progression and mortality of patients. We discussed the design of two distinct types of AI imaging biomarkers, a supervised radiomics biomarker, and a weakly unsupervised GAN radiomics biomarker. We presented results indicating that these AI imaging biomarkers outperform currently established prognostic biomarkers in predicting the overall survival and risk stratification of patients with idiopathic pulmonary fibrosis or the COVID-19. Therefore, these AI imaging biomarkers have the potential to provide effective image-based prognostic biomarkers to be used as a clinical decision aid for the management of patients. Thank you for your attention.
Thank you, Professor Yoshida, for your exciting talk. We have received a number of questions from the participants, which indicate that how interesting is your talk. So what we will be doing, we will be sending all these questions to Professor Yoshida, and as soon as we receive the reply from him, we will upload those reply to the YouTube channel of Spark. So thank you once again for your exciting talk. Thank you very much. Now our next speaker is Professor Annemiek Geluk, and she is from Leiden, University of Leiden, Netherlands. So I would like to briefly introduce Professor Geluk with the participants, and his biographical bio sketch is briefly like this. Professor Annemiek Geluk is the chair professor on immunodiagnostic and mycobacterial disease at the Department of Infectious Disease in the University of Leiden. She has wide experience in industry, and her research is focused on the immunology of leprosy and tuberculosis, particularly the identification of human T cells that are essential to protective host immunity or immunopathology, including identification of immune, genetic, metabolic of the host biomarker. As a PI, she has received large grant on leprosy endemic area, and Professor Geluk is the advisor of multiple leprosy and TB research projects abroad and co-authored more than 130 international peer-reviewed scientific articles. Thank you very much, and now let me welcome Professor Geluk for the next series of lectures. Thank you.
Can you hear me?
Yeah, please go ahead. We are not getting your camera view. Hello?
Yes, hello.
Professor Geluk, we are not getting your camera view. Actually, it's just a blurred image. Yes, right. Please go ahead.
Can you also hear me?
Yeah, I can see you. We are getting your voice properly, video properly, as well as the screen is shared now. You may please go ahead.
Yeah, I already shared my screen.
Yeah, we are getting.
We have your shared screen.
All right, okay. Thank you very much. So good afternoon in India, so good morning here in the Netherlands. I Can I hear myself twice? Can you mute yourself? Because otherwise yeah, okay, thank you. So thank you for the invitation to share some of the work that my research group in the Netherlands is doing on the mycobacterial infections, leprosy and tuberculosis. So today I would like to share with you some research advances in the point of care test that we are developing for leprosy and TB. So first of all, at the LUMC, so at the Leiden University Medical Center in Leiden in the Netherlands, we have a huge leprosy and tuberculosis research group. We focus our work on the development of vaccines and clinical drugs for tuberculosis, but also a lot on basic research. So that is mycobacterial research, immunology on mycobacterial Mycobacterium leprae and Mycobacterium tuberculosis. But today I would like to focus only on the diagnostics, so the development of diagnostic tests. So first, some background information. You may be aware of that leprosy is caused by Mycobacterium leprae, and that it is the first bacterium that was identified to be associated with human disease. Even before Mycobacterium tuberculosis was shown to be causing tuberculosis. So actually, the worldwide prevalence has decreased a lot since the 80s due to the introduction of MDT, so multi-drug therapy, which is a combination of 3 antibiotics. It ranks second as a disease in humans caused by mycobacteria, so after Mycobacterium tuberculosis. However, leprosy is not as contagious as TB is. And even in endemic countries, so in the hotspots where it's still occurring now, it's not very contagious. So you need a huge doses of the bacterium to get infected and also to develop the disease. So if we look at the incidence of leprosy now, India has the largest incidence of leprosy patients, about 60% of the global number. And after that, Brazil and Indonesia are second and third. And there are, however, a lot of countries still that are reporting hotspots. So that means that there are not endemic anymore if you look at the total figures, but in these hotspots, there are a lot of leprosy cases. However, because the country is not endemic anymore, the interest to identify and to treat leprosy has been decreased a lot. So therefore, there are still some challenges that we're facing in leprosy research. Number one is that there is a stable new case incidence globally of about 200,000 per year, so meaning about 23 patients per hour. And 10% of those individuals are children. So this means that there is still transmission ongoing. And if we don't stop that, then we will not be able to finally eradicate the disease. Furthermore, asymptomatic infected individuals, they may be a huge source of transmission because they're not identified and they still don't have clinical symptoms, but they can transmit the bacterium. And then, like also with COVID-19 nowadays, that people can still be asymptomatic, but they can transmit the pathogen to others. So therefore, it's important to early diagnose the infected individuals and to further prevent handicaps and also to interrupt transmission. However, at this stage, like also with other diseases, there are no good diagnostic tests yet that can detect infected individuals without any symptoms. So this is where at the LMC we are working on. So if you have Mycobacterium leprae then most of the individuals are able to kill the mycobacterium, and they don't develop disease, and they don't carry the bacterium with them, so they're not contagious anymore. However, a small percentage of the individuals, then in those individuals, lepre can survive, and it can be there for like 2 to 6 years, but sometimes even over 10 years or 20 years. And that those individuals, they can potentially also be contagious. So they can transmit the bacteria, and then the cycle will go on. So what we are aiming at is to identify those individuals that are infected, are possibly going to develop the disease themselves, but are also where the disease is not endemic. And we identify biomarkers for those that are clinically shown to have either leprosy or TB, and for those that are infected only. And we then compare the biomarkers for those groups using clinical information first in this in this research, first as a golden standard. And I hope this works. So what we are aiming at is to identify biomarkers that are based on immunology, because if you get infected by either a virus or a bacteria, mycobacteria, also fungi or helminths, then the invasion brings about a reaction, and the response is focused at elimination of the pathogens. And this could be like T cells, B cells, macrophages, and all those cells that are fighting the elimination of the pathogens. They are producing different biomarkers in the host. And the biomarkers can be identified. And this is, for leprosy, very important, because for leprosy, it's not that one biomarker fits all. So that one biomarker can be specific for both those that are post-bacillary and those that are multibacillary, because there is a clinical spectrum for leprosy. And this is very characteristic. It may also be present in other diseases. It has already been shown to be slightly there also for TB, but in leprosy, it is very prominently present, because on one hand, people that are so-called promatous leprosy patients, they have a lot of bacteria, so they're called multibacillary patients. And this is what you see on the right here in this slide. So they have no T-cell response, so no good protective cellular mediated immune response. And they have a lot of bacteria, and also they have a lot of antibody response. And on the other hand of the spectrum, there are tuberculous leprosy patients. Those are post-bacillary patients. They don't have a lot of bacteria, and they do have a good protective response, but just a little bit too much, so that they also cause damage to their own skin or own nerves. So for the context, for those that are not yet present as identified as patient, there is a mixed biomarker patterns, and therefore we need to have longitudinal studies following those individuals in time to see who is developing disease and which biomarkers can be predictive of development of disease. So this is what we have been doing in my research group. And first of all, I want to inform you that there are host serum markers, so biomarkers based on antibodies directed against PGL1, for instance. PGL1 is a component of the cell wall of Mycobacterium leprae that is really very unique for Mycobacterium leprae. And the individuals that have multibacillary disease, they have a lot of antibodies also. And we have shown, and others have shown that as well, that there is a correlation between the infectious load, so the bacillary load, BI, and the PGL1 response. So what you can see here on the right is that those that have a BI that is positive also have a high PGL1 and anti-PGL1 response. However, the PGL1 response does not indicate that the individual is a leprosy patient, nor is it so that the PGL1 positivity indicates prediction. So we have done longitudinal studies in Bangladesh assessing those individuals that were PGL1 positive, and we found no correlation between PGL positivity and development of disease. However, those that are high PGL1 positive usually are developing disease and to the multibacillary component. So to identify biomarkers that are also important to diagnose the post-bacillary side of the disease, we are focusing on cellular biomarkers. And we have done so in large studies since 2012 already in Bangladesh, for instance. So these are field studies in which we go to rural areas where leprosy is still highly endemic. We take blood of individuals, incubate the blood with Mycobacterium leprae antigens overnight, and then analyze the supernatant of those whole blood assay by multiplex cytokine analysis. So this allows us to identify biomarkers that are specific for leprosy patients, to identify the differences between the cellular markers in multibacillary and in post-bacillary patients, and also identify the response to, for instance, BCG vaccination, or the response in healthy household contexts of leprosy patients. And so in addition to that, we are also working on the identification of genetic biomarkers, and we use transcriptomic analysis for that. So for instance, qPCR, RNA-seq, and MLPA. So what we have found for the proteomics part, so this is based on the Luminex, so the multiplex serum protein analysis. What we have found that there are two ways actually to identify proteins in serum that are useful as a biomarker. So either directly in medium or after overnight stimulation. So when you want to have a rapid test, you of course don't want to have a 24-hour incubation step. So if you focus on the upper panel, there you can see where it's indicated in red that there are biomarkers that are different between the two groups. For instance, the multibacillary patients and the endemic controls. So EC stands for endemic controls. You see that it is highly red. This means that the number of biomarkers that are different between those two groups is large. However, it's much more difficult to identify biomarkers that are different in multibacillary patients versus post-bacillary patients. Hence, you see the yellow squares indicating that there are less biomarkers difference between those individuals. So, however, if you have an overnight stimulation step, then you see that there are more biomarkers that are different between the endemic control group and, for instance, the multibacillary patient group. So what we then did is we looked at what would be the added value besides having an antibody biomarker and then using the cellular biomarker. And what you can see in this is that the percentage of identified patients is about 32 to 36% higher if you use cellular biomarkers in addition to the anti-PGL1. So having identified those biomarkers, now it was the challenge to move from high-tech to a low complexity, to be able to use the biomarkers also in the field. So for that, we use the UCP reporter technology. The UCP stands for upconverting phosphor nanoparticles, and it's actually a field-friendly alternative for just an ELISA. It is very important to realize that this is a quantitative assay. So it's not like plus or minus. It's not dependent on who is doing the test. So it's not operator dependent because it's the quantitative assay and there's direct correlation between the signal and the amount of biomarker. It's very sensitive because there's no background. And it's also stable, so it can be used without a cold chain. It can be used in the field at higher temperatures. The only thing it's a little bit sensitive for is humidity, but if you use like silica packs, then it can also be used in the field and be kept there. It doesn't bleach, so it gives you a permanent record. So the strip, when you have analyzed it, you can use it directly after you've done the test, but also when you keep it for about a year, you can reanalyze it again and again. So this is what it schematically looks like, the UCP lateral flow for one biomarker. So you add to the sample pad, you add a little bit of blood or blood in buffer, and then it flows the right direction. And then it first encounters the test line where there is the capture antibody for the component that you want to, the biomarker that you want to analyze. And then it has the control line to see whether the test has flowed correctly to the absorbent pads. So this is what you see when there is a positive test, a negative test. So then the test line here does not contain any UCP label. There's only a flow control label. And this is what you see when there is both a test line of positivity and flow control positivity. So then it means that there on the test line, the biomarker has bound, and then the UCP biomarker has again bound to the test line. And this slide is a little bit more schematic. You see, for instance, this is a UCP lateral flow test protein, which is specific for IL-10. So these are the antibodies that are labeled with the UCP nanoparticles, and they can bind IL-10 in them in the sample, and then IL-10 bound UCP labeled antibody can again bind to the other antibody, which is on the test strip, so that you have a sandwich, but then on a lateral flow strip. So it is a lateral flow, which is cheap, which is low complexity, but then using the high-tech UCP particles on it. So this is what it looks like, actually. So these are the strips, the way they look currently. This is the whole blood assay. And then you either get your serum or you get your stimulated whole blood. And these are then a lot of strips that we analyzed. And we analyzed them in a so-called UCP reader, which is a reader which is specifically, so it's custom made for the UCP and gives a readout here. But it's a portable reader, so you can use it in the field and you can later on connect it again to the to a laptop to store your data. So we recently published some more. So if you are interested, you can look at the e-biomedicine paper by Van Hooy et al., which gives you an idea of which biomarkers we are now using and which biomarkers are useful and for what purpose. So for identifying infected individuals or identifying patients or to make a discrimination between MB and PB patients. What I want to add to this is that if you have 5 biomarker signature, then the area on the curve, so the sensitivity and specificity is better. So although we have been using single UCP lateral flow strips, it's still better to have a combination to get more higher sensitivity and specificity, so-called biomarker signature. And this is again what we're working on now. So I mentioned that for leprosy field work, you need low complexity. And of course, taking serum will require also a phlebotomist. So you have your venous blood. Well, if you want to avoid that and only want to have your finger stick prick, this is also also possible now. So we have the UCP now also available in a cassette for anti-PGL1 to work directly with the blood. So we collect, to maintain the quantitative aspect, we collected the blood from the finger stick with a 20 microliter minifet, which exactly has that amount. And we know that UCP is not hindered by hemolysis. So the hemoglobin in blood does not interfere with the UCP signal. So we have that in stock now as well for anti-PGL1 antibodies, but we're working on it for other cytokines and chemokines as well. So to give you an idea what we are using our test now is the applications, for instance, for contact screening. So on the left, you see an image of work in Bangladesh or in China, also in Brazil and in Nepal. We use it for monitoring treatment efficacy, so to estimate the biomarkers on diagnosis and also first treatment. And we have also been able to apply our PGL1 test to animals, because you may have known that there are animals in the UK, the squirrels, the red squirrels, that have been infected with Mycobacterium leprae. So to conclude, this is an overview of our TB and leprosy work. I've only discussed now, because of time, only the leprosy work, but we're also working on TB with a similar lateral flow assays. So I'd like to thank you for your attention. And here I show all the individuals and the groups that we've been working with in both in the Netherlands, but also in Brazil, in the United States, Ethiopia, Africa, China, Bangladesh, Edinburgh. I thank you for your attention. And if there are any questions, please let me know.
Thank you. Thank you, Professor Geluk, for your very interesting talk. We have received a number of questions for you. Hello, are you there?
Yes.
Yes. Can I place the questions one by one, and you will be addressing those questions? Then I can ask. I can tell you the questions which are there, and you can. Okay. Thank you. So the first question, which I'm telling, there are a number of questions. I have selected few of the among this huge number. So first question I'm asking you, that microscopy-based imaging for detecting TB from stained sputum smear is well known. Any such test for leprosy? What is the difference in the structure between Mycobacterium causing TB and the one causing leprosy?
Well, thank you for the question. So if I repeat it, so to make sure I understand it correctly, what we're asking is, what is the difference between Mycobacterium leprae and Mycobacterium tuberculosis?
Yes.
They
are asking that structural difference between Mycobacterium causing TB and the one which is causing leprosy.
This is not my field, though, but as you know, in around 2000, both the structure or the genome of Mycobacterium leprae and Mycobacterium tuberculosis were identified, and what was very clear is that Mycobacterium leprae has a lot of pseudogenes, and there was a lot of degeneration of the number of genes compared to Mycobacterium tuberculosis. Therefore, Mycobacterium leprae requires a host to survive. So it can't survive without a host. So whether it's an animal host or whether it's human host. And this is different from Mycobacterium tuberculosis, which is much stronger, can also be cultured in the laboratory. And so I think I have to leave it at that with respect to the structure. They both are very similar. And if you look at the lineage, so the lineage tree in which they are, they're very close together with other mycobacteria that are also slow growers. So I hope I have addressed this sufficiently.
Yes. So the second question is UCP LFA based on quantum dots?
UCP, I may go back to that. So UCP stands for upconverting phosphor nanoparticles. So it is based on rare earth. So these are structures and that are not present normally in the environment, and therefore they have a low background. Yeah. So rare earths have a crystal structure, and if you put infrared light on the crystal structure, it will emit light with a higher energy. And this is then picked up as the signal in the UCP reader. So therefore it's also called upconverting, because it converts light with a lower energy to light with a higher energy, because it goes from infrared light to the visible spectrum.
Okay. The next question is, how far is nanotechnology successful in diagnosis of treatment of mycobacterial infection?
So can you repeat the question?
How far is nanotechnology successful in diagnosis of treatment of mycobacterial infection?
So the UCP lateral flow, how successful it is already in diagnosis of leprosy? That is the question.
Yes.
Well, as I've showed here in my presentation, what we have been doing is identifying Mycobacterium leprae infected individuals. So what we can already do is identify infection and also identify it in a quantitative way, meaning that we know the extent of infection. So therefore, we can identify those that need treatment, but also we can monitor disease treatments, whether that is efficient. And because we measure not one biomarker, but because we measure multiple biomarkers, we can use those biomarker results, the quantitative results, also to differentiate between people that have disease and people that have infection only.
Okay, there are plenty of other questions also. So what I will request you, so if you agree, then we can send you the questions which has come from the participant, and if you can address those questions and if you send it back, we can upload it in the YouTube channel of our Spark. So that will, I think that will, there are plenty of other questions. So with this, I want to extend our heartiest thanks on behalf of the entire Spark team to you for your this nice and exciting talk. for this particular conference. And thank you for coming and giving this talk. Thank you.
Thank you for inviting me. It was a pleasure. Thank you.
Thank you. So now I would like to inform the participants that we will have a tea break. So after this tea break, once again we will assemble here at 15 sharp 15:50. So now there will be a break. Thank you.
Change of place has a problem.
But system one is good. And why don't you try with coconut oil? Coconut oil is very good. I think coconut oil is very good.
But the taste.
Professor Ramakrishnan.
Yeah. You want me to log off?
Yeah, sir. Actually, by mistake, the host has been assigned
No
to
problem.
your
No
this.
problem. I will
Okay,
just.
sir. Sorry for the inconvenience, sir.
No problem. We
Okay.
understand. Hello. You are unable? Hello? Hello?
Yes, sir, I can hear you.
Yeah. What is that?
You can switch on the camera and you can just share the presentation once.
Right? Able to
Yes,
see
yes.
me?
Yeah, we can see you, sir. And can you share the presentation once, sir?
Able to see?
Not yet, sir. Yes.
Now?
Yes, we got it. Overview of respiratory system.
Is it okay?
Yes, it's okay. You can stop the presentation once the session starts, then only you can share, sir. Okay, camera, let it be on, sir.
Okay.
Sir, we are still getting your presentation.
It's possible. Now?
Now presentation is stopped. So let it be in this mode.
Video also off?
Yes. Now it's perfectly fine, sir.
Video switch off?
No, video, let it be on, sir. Okay.
That's good.
I checked it.
Good afternoon, everybody. Our next speaker is Professor Ramakrishnan S. Let me give a brief introduction regarding him. Dr. Ramakrishnan is a professor of biomedical engineering and also the head of the Department of Applied Mechanics in Indian Institute of Technology, Madras. He had his initial research training at RWTH, Chennai during 1995 to 97 in the area of biomedical image processing. His current interests include medical informatics, device design, calibration, emotion detection, and standards and regulation. He leads a group of 15 researchers work in this area and also has graduated more than 20 scholars. He has published over 250 papers in journal and conferences. He is a founder director for 2 spin-off startups and is a sought after international research collaborator. Now I greet Dr. Ramakrishnan to deliver his lecture. Let us listen.
Can you share? Okay.
The screen is not coming fully. Can you just?
It's okay?
Yes, sir. Right now. You can even go for the full screen, sir.
Are you able to see?
Yes, sir. We can see, but you may actually give. Yeah, right now. Okay.
Okay?
Yes, sir.
Can I proceed?
Yes, please, sir.
First of all, I would like to thank the organizer, especially my nice colleague, Sir Manjunath, for giving me this opportunity to discuss some important issues in this very nicely organized this webinar conference, despite all odds. Yeah, I'm very happy to be part of it. I just wanted to discuss today on enhancing the diagnostic relevance of imaging respiratory conditions, challenges and approaches. And as I was introduced, I'm a professor in biomedical engineering. So I'll start with briefly because this imaging respiratory system is a key field in itself. Very dynamic changes are happening. So I would like to give a simple overview of what we could do, what future directions we will have in terms of research and the need. With this, I would like to start. If you look at the respiratory system imaging.
Professor Ramkrishnan, can you hear me?
You tell everybody to switch off the mic.
Please, everybody, switch off your microphone. It is causing lots of disturbance. We are not able to hear Professor Ramkrishnan properly. And Professor Ramkrishnan, you switch on your microphone and camera, and please little bit away from the microphone because lots of echo is there.
Okay. Now is it okay?
Yeah, it's okay. Yeah, it's okay.
Now it is okay?
Please on the camera, please. Please switch on the camera, please.
So I don't have the video. Is it okay? Is it okay?
Sir, I think you have lost your presentation. Can you?
Yeah.
We are not getting the presentation right now. We are just getting a black screen. So can you start the presentation once again, sir? Earlier it was coming perfectly, only the camera view was not there.
Getting now?
Sir, you just stop the presentation once and restart it, sir. I think that will solve the problem.
First shop, huh?
Stop the presentation once again, and now you switch on your camera and start presentation, sharing the screen.
Okay.
Sir, your video has come back. Yes, right now it's perfectly okay, sir. Please go ahead.
You're not getting it?
Yes, yes, we are getting your video, audio as well as the presentation. Please go ahead, sir.
Okay.
Okay.
Is it okay? Is it fine?
It's fine, sir. Please go ahead.
I can move ahead, right? Yes, yeah.
Okay.
So, I will just briefly look at what imaging part of imaging is happening on the respiratory system. It essentially comprises of analyzing the thoracic cage, the lungs, and diaphragm. So, analysis of this kind of system involves lung function analysis, both invasive and non-invasive tests, and passive and active tests, and molecular analysis of breath usage and respiratory analysis. Primarily, imaging methods for respiratory disorders include the microscopy, bronchoscopy, and radiography. So now, if you look at the role of imaging in respiratory things, what our interest would be to look at the changes that are happening in the lung area, structural changes in airways and pulmonary vasculatures, the loss of pulmonary tissues, especially that are causing some changes in textural patterns in images, and inflammation of lungs, and decreased airflow in alveolar surface area, and enlargement of mediastinum, enlargement of central mediastinum, and lung folds, and facilitate visualization of organs, typically lungs during perfusion and visualization during perfusion and ventilation. So these are all the functions primarily the imaging is used. So now, diagnosis and management of respiratory disorders, look at the imaging options. What are the imaging options we have? Is that these are all the frequently employed. Starts pulmonary this chest radiographs, kind of radiograph. Then comes your computed tomography, and then magnetic resonance imaging. So then you have the lung ultrasounds. Now ultrasound, lung ultrasound has become, secondary to chest X-rays, and we have positive emission tomography. These are all modalities that are being now used to analyze changes that are happening with the imaging modalities that are available now and being widely used for the analysis of respiratory functions. So if you look at the chest radiographs in for respiratory functions, is one of the earliest forms that's being used. See a lot of noise. Am I audible? Yeah, am I audible? Unable? Am I audible? Hello? Am I audible?
All the participants, please switch off your mic and video. There is a lot of disturbance. All the participants, please switch off your mic and video. Yeah, Professor Ramakrishnan, you can go ahead.
Yeah, please. Chest radiography is essentially testing for the initial test for the abnormalities in the respiratory function. It's primarily an ionization radiation, and it works on the 2D production. Widely used everywhere, even in the primary category, it's available everywhere. It's affordable and non-invasive, and very less radiation doses used. It's primarily suitable for mass screening, and they suggest this pneumonia and COVID-19, tuberculosis, and fungal colonization of pulmonary cavities. So the next commonly used is the computed tomography, and this is a gold standard for diagnosis of infections in the pulmonary systems. There are multiple projections of an X-ray, essentially an ionized radiation. It offers much superior spatial and contrast spatial resolution and contrast resolutions. So probably some subtle variations we can extract out of these images. It also facilitates the diagnosis of a fungal colonization, tuberculosis, emphysema, and COVID-19. So now the third one is this magnetic resonance imaging, and you can see here there's some representative cheap-weighted images are shown here. And principally it works on the strong magnetic field and radio waves to generate images of the organs. So it's radiation free, provides better performance for visualization of structural and functional radiations, especially of soft tissues where X-rays cannot diagnose. So understanding pathologies and the mechanisms of pulmonary infection useful in evaluation the pulmonary infection. But long time scanning time and high contrast compared to CT and US, that's one of the disadvantages of using this. So positron emission tomography is the next one being used for COVID also. It uses radioactive substance to visualize and visualize the metabolic processes, measure metabolic processes. This used in the detection of lung density abnormalities, glucose metabolism, and monitoring of lung cancer. It also measures the strain imposed by mechanical ventilation on different parts of the lung. It's an advantage over other methods. It provides functional information of on acquired respiratory distress syndrome and ventilator-induced lung injury, and chronic obstructive pulmonary disease, COPD. So now look at, there are the five these things. If you look at what are the issues and challenges currently we are using imaging system for assessing respiratory disorders. Look at, as we saw that X-ray, MR, ultrasound, and PET. If you compare all these imaging modalities, many of them are modalities are used separately. And if you look at X-rays, they have the low resolution structure, resolution is low, it has high inter-observable variability, and there is a danger of radiation exposure, and additional tests ought to be performed to confirm the diagnostics. If you look at the CT, which is again derived from the X-rays, projected X-rays, it has limited functional details, dynamic informations are not present, and radiation dose is very high when we compare them with X-rays, and cannot be used for routine use because of some logistics and other issues. Look, MR is we can image soft organs. It's a low spectral resolution and provide generates a lot of artifacts, react tissue interface, and lengthy acquisition time. And, ultrasound, as I said, ultrasound is only next to X-rays. Is most commonly available, affordable, portable, and cannot get built over inflation. It's a poor presentation. It's difficult to examine these patients. It requires expert knowledge. So inter-observer variability is very high. Now, you take a bed, I think you get lots of metabolic information in this with very poor temporal resolution. Again, variability is very high, very expensive. Logistics is involved, and so these are all some of the challenges. Look at current pandemic issues. If you look at COVID-19 and imaging biomarkers, there are different kinds of reports, contrary reports, different observations have been reported if you look at the literature. And essentially a respiratory tract infection caused by this virus, and X-rays and CTs are widely used everywhere. There are imaging features. If you look at the imaging features, and the patchy areas of ground glass opacity, you can see here patchy areas of ground glass opacity, and consolidation is done predominantly in the periphery of the lungs, characteristics of manifestation. What are the problems in this thing is that overlapping of biomarkers with pneumonias of different regions. There are overlapping, is a major issue now to differentiate this pneumonia from this kind of virus infects induced by virus. So this can be negative even for the few days of symptomatic infection. These are all the advantages of using imaging biomarkers currently. Look at the strength and challenges of this in the under prevailing conditions of COVID. X-ray, yes, it's fine, easy to perform, portable, but may be negative in initial stage and low specificity and sensitivity. If you look at the CT, the optical structural information used to follow infection studies, but limited high radiation dose. So the ultrasound, and again you have only you can use it for prevention. Then MRI, you have non-invasive method, but alternative estimate of lung lesions only that you need the lung volume information, so you can use it. But as I said, lots of you need auxiliary diagnostic tools for this. So what is now? What are all the challenges of diagnosis and management of respiratory disorder with COVID-19? First of all, identification of asymptomatic cases using imaging is difficult. Yes, there are not indicative in asymptomatic cases, even those with mild symptoms. That is the first one. And other vital pneumonia, you cannot distinguish other vital pneumonia with similar kind of image characteristics are found in COVID-19. Manual diagnosis requires lots of labor. The specific imaging protocols are not still available, and there are large database. What we need for AI and other things are not available now. There's a very high risk of the viral exposure and the technicians and front-end healthcare workers is a possible. So these are all the challenges. So what could be the potential approaches? what potential approaches in imaging respiratory? This could be multimodal imaging. So far, they are using only single mode. There could be emerging imaging modalities, new modalities. We had to come up with new imaging modalities, and we can use artificial intelligence and radiomics, and we can use informatics, medical image informatics, and advancements. Then we have precision medicine and interventive analytics. These could be the new approaches. These are all some of the new approaches that could be used with the respiratory current prevailing conditions to analyze the respiratory disorders. Multimodal imaging, there are different types of. For lack of time, I'll just go too fast and share it. You can send a mail if you want anything. So multimodal imaging is that there are three different types of multimodal imaging. SPECT, CT, PET, MRN, all the three, though it is still not being used now for COVID, but could be very effective because you can, both the things, you will have, if you have a technology, we have to generate some technology for simultaneous acquisition and automatic imaging. If you have that, you'll get both metabolic, structural and functional information so that we can make better these things. There are future trends that are coming up in imaging displays, electrical impedance tomography is now again coming up, optical coherence OCT, and then hyperpolarized MRI, new protocol, and phase contrast X-rays. These things are emerging modalities, and in addition to multimodal things, we can also generate information from this. The thing is how artificial intelligence and radiomics in this field, like if you look at that, this is a it comes as a complete package. You have this process involves image acquisition and segmentation. The different software tools are now available. Then feature extraction using histogram based, geometry based, and texture based, and topographic analysis. You can bring all the image into data domain, convert all the image features into data domain. Then you can analyze using machine learning methods. There are different this seems to be a potential area of research where you can use even with, especially if you take the deep learning methods, even with limited data sets, people are coming further, so that you can extract useful information, biomarkers from the imaging. This is one of the this thing. Then if you look at the informatics point of it, unfortunately, this informatics, what are all the advantages that are happening in communication has not been brought into medicine. That's one of the, I don't know why, because all advancements only happens in medical field, but this kind of imaging is still many of most recent advancements in informatics have not been tried out. So now what we do is the real-time monitoring of imaging, image acquisition, and continuous monitoring and multimodal data fusion in real time, and integration of legal data records. All these things could be, the medical informatics community can help us to get the next this thing. Look at the real-time monitoring and wearables. This is health monitoring systems. They automate patient monitoring tasks and improve efficiency in clinical testing. So, for example, in this case, we can have a thermal imaging. You can continuously have thermal imaging in terms of COVID-19 or related respiratory disorders. You can also have multimodal image fusion or data fusion or decision fusion. You can take signals from all the associated things. You can also use your gateway and medical servers and generate a database. You can share it with the users. So you can have a dedicated protocols and communication schemes for us, right? You can have a patch electrodes. You can also measure and image respiratory disorders. And you can come up with some applications. Image fusion is a most recent thing. Image fusion is not image fusion and decision fusion is also now being used. So because multiple algorithms are used, multiple machines are fused, and you can come out with better medical imaging, better decision. You can come out with better decision based on the image features. You can see the features are extracted, and then you can use MRI and PET or the MRI and SPECT. So multimodal features, multimodal features are fused. So even though sometimes they are not simultaneously recorded, you can use these algorithms, and then we can one can come out with better decision in these things. And finally, so I would like to say that this most recent advancement is that this IoT based interoperable electronic health records is not quite right so far. There where you have from diagnostic laboratory, you can generate, you can have multiple access, generation loss by extracting useful image features. Either you can do the processing here, you can share in the data domain, or you can share maybe in the image domain itself. We have lots of data points where you can health history of the information can also be shared. Informed decision to be made. This could be the advantage of using this. And another thing I think is this precision medicine. The precision medicine is what still now it will be very useful, especially for respiratory system disorders. You have you can have a individual you can have individual treatment modalities for individuals, whereas you can have a target group instead of having target groups. You can have the individual persons. You can use all the data like from your demographics, lifestyles, food histories, medical records, compliance, and other things. So this is also an advantage which still this imaging community has not exploited this. This can also be done so that finally, I would like to conclude this is interactive analytics. It's also now coming out where we have a current clinical data is passed on to your this thing. You have solutions for precisely dedicating areas of your own interest and multiple image acquisitions and the delineation of your area of interest, extraction of relevant features, all domains could be put together, and quantitative and qualitative image analysis can be made. And finally, we can come up with either precision or target. So this is other advantage of this. Conclude with, and let us try to there are some issues and challenges imaging respiratory, especially we don't have faster acquisitions and low. We need solutions for low radiation dose. So precise anatomical imaging methods are required. So then variations in imaging parameters, different slice thickness, solution, etc. And there are predominant amount of imaging data for storage and automation. There are multiple modes also. So conventional method used for respiratory disorders, so if they alone they cannot provide biomarkers for us for the informed decision. So what what could be the future intensive research approaches could be your multimodal imaging as we discussed, and emerging imaging modalities, new modalities based on CTs and medical imaging informatic advantages, real-time acquisition, transmission, and internet-based things, and precision medicine integrated analytics. I'm pretty sure these are new approaches. Probably we can come out with better extraction, better information, and biomarkers. So with this, I would like to conclude. I'll be very happy to have share anything, and you can also reach to. Thank you. Hello. Yeah.
Thank you, Professor Ramakrishnan. There are few questions which has been directed to you. Among this, I am going to ask you two questions, and the other questions I will be forwarding it to you, and which you can answer and which you will upload in our Spark YouTube. The first question is, how far the imaging technique has reached in diagnosing COVID-19?
Yeah, as I said, there are two major challenges is that there is availability of, we cannot have the availability for sophisticated equipments, and then another thing, we cannot have functional analysis, we can have only statistics. It's the differentiation of normal pneumonia and your COVID-19 kind of things. They pose the same kind of imaging features. That is still a challenge that has not been fully addressed so far. So people people are trying everywhere. It's still a challenge. I would say that we are not totally successful in these things.
Okay, the second question is FDG PET useful in the diagnosis of COVID-19?
It was
Is FDG PET useful in the diagnosis of COVID-19?
I think, because you can see some more this thing, but it's been much tried out. But you can expect some, because of metabolic effects and could be extract some information could be extracted. But I have not come across anything, and it's not my area. But I understand that it is possible.
Okay. Thank you for your informative lecture. Thank you, Professor Ramakrishnan.
Thank you. Thank you, Manjunath, Professor Manjunath.
Good afternoon to our viewers and good morning to Dr. Sudeepa Bhattacharya. I think so it's early morning in America. So our next speaker is Dr. Sudeepa Bhattacharya. Let me introduce her first. Dr. Bhattacharya is currently an Associate Professor of Bioinformatics and Data Science in Arkansas State University, Arkansas, USA. After completing her bachelor and master degree from Calcutta University in chemistry and biochemistry respectively, she obtained a second master degree in bioinformatics under the mentorship of world-renowned bioinformatician Dr. David Wishart in the University of Alberta, Canada. As bioinformatician, her primary research interest is in understanding disease and toxicity related pathophysiology using a system biology approach, integrating complex data from multimodal experiments, developing graph theory based network model to understand underlying biological mechanism. Her overall journal publication has an average impact factor of 7.012, including top journal, for example, PNAS, Alzheimer's and dementia, cancer research, clinical cancer research, Journal of Bone and Mineral Research, etc. Now I invite Dr. Sudeepa Bhattacharya to deliver the lecture.
Can you all hear me? Hello? Can you all hear me?
Yes, madam, we can hear you. We can see your presentation slides.
Oh, you can see my presentation? I cannot hear you properly.
Yes, ma'am. We can see the presentation. We can see your view. Audio is perfectly okay. You may please go ahead.
Okay, and let me see if I can go to the presentation. Now, you can hear my voice clearly, right? I'm hearing some echo back from you, so if you all can mute the microphone, that would be very helpful. And okay, so I'll just get started. Hopefully you all can hear me. And so talk today is on data-driven modeling for precision medicine, a case study in major depression. So here is an outline of my talk. I'll start with a brief introduction, then major depressive disorder impact during this pandemic of COVID-19. I'll talk about the major challenges of the disease, how we are addressing some of these challenges in the through our research work. We work in a consortium mode. I'm actually attached with two large consortium. One of them is the Mood Disorders Precision Medicine Consortium. And we are about to complete the phase 1, and I'm going to talk about some of the research that I or my group has participated in the phase 1 of this consortium. And lastly, if time permits, I'll talk about some of our future work. There's a lot of noise in the background. Is any way, can that noise be muted a little bit? So, major depressive disorder, here are some of the general facts. So, it is a leading cause of disability and mortality, and it affects about 16% of the US population. 16% is around more than 50 million people in the US sometime in their lives. And MDD associated with high rates of unemployment, impaired work productivity, family dysfunction, cardiovascular disease. It comes as a comorbidity in a number of diseases. And of course, it comes with a high rate of suicide. Now, MDD or depression is disorder, but rather a very complex syndrome. It comes with multiple types of involvement in the central nervous system disruptions. For example, sleep disturbance, appetite, then feeling sad or not pleasant, then cognitive disabilities and on. And it comes with a wide range of clinical presentations, courses of illness, and we don't know much about its biological underpinnings. It's a very heterogeneous disease, and that explains why we are not able to target our treatments. There are not treatments that can treat all the patients. It has a high resistance rate to treatment. Many people do not respond to treatment at all. And then we are, at this point in time, unable to develop preventive interventions, predict longer-term outcomes, and on and on. So this is a United Nations policy brief during COVID-19, and I just want to highlight 2 points here. That the global economy loses more than $1 trillion per year due to depression and anxiety. And it affects around 264 million people in the world. And actually the number is kind of more. I mean, according to our data, it's around 300 million. And around half of all mental health conditions start very early in the at a very early age, around 14. And suicide is the second leading cause of death in young adults between 15 and 29 years. So it's, but the disease mental health is by itself a pandemic. And here you can see the map of United States, the different states reporting symptoms of anxiety or depressive disorder during the COVID-19 pandemic. And you can see the colored states here, which reported the lowest percentage. Even there, the percentage of population suffering from this disease is 25 to 28%. And on the right-hand side, you can see the trend plot that shows that from April to the end of June, the level of anxiety and depression has remained pretty steady in all the 50 states of the US. And now it's even showing an upward trajectory. So nearly half of the US adults are reporting mental health related issues at this point in time. So major challenges of NTD. We have no reliable means by which to select which treatment will work for which patient. Clinicians today rely on a trial and error approach to find the best treatment for each person. And we don't understand the biological underpinnings of the disease or why some patients respond to the drug and treatment and why do not. And we also have limited ability to predict depressive episode recurrence or relapse after recovery. And we found that neuroimaging analysis alone, often the genetic analysis alone, do not yield reliable diagnostic markers for the disease or its subtypes. In fact, the disease is reported to be polygenic, and so these SNPs contribute, by small amounts to the disease. So now, our mood disorders precision medicine consortium, I joined this consortium about 3.5 years back. It was started by the researchers in Duke Medical Center and Mayo Clinic, Duke and Schulman Group in Mayo Clinic. And we are now completing, so it's in existence for about 5.5 years. And we are now completing the phase 1 and going into phase 2. We have expanded. We are now Max Planck from Germany, Oxford University from UK, the Netherlands, the Nesta group from Netherlands involved, along with Caltech, Circus Mesmanians group, and then Rob Knight's gut microbiome group from UCSD are also involved in the phase 2. So I'm going to talk about 2 studies that I have mainly participated in the phase 1 of the consortium. So one of the cohort came from Mayo Clinic, the Mayo PGR AMS cohort, and then another cohort came from the Emory, the Emory Predict cohort. Now, on the left, you can see the Mayo cohort. The Mayo PGR and AMS cohort, it consisted of about 290 unipolar patients with major depressive disorder. And these patients were profiled at baseline, 4 and 8 weeks of drug treatment. And they were treated with the SSRI antidepressant, citalopram. And then their depression severity was assessed by a rating scale called HAM-D rating scale. Now, the Emory Predict cohort, it came from the clinical trial, Predict clinical trial. It consisted of 3 arms, 2 drug arms, citalopram, duloxetine, these are antidepressants, and then CBT, cognitive behavioral therapy, which is a drug-free arm. So each arm consisted of about 100 patients, MDD patients, who were also again profiled at baseline and multiple time points up to 24 weeks post-treatment. And depressive symptom severity were again assessed by rating scales, HAM-D, and also QIDS. I will concentrate mainly on the results we got from these 2 studies. So what was our approach? So we collected lots of metabolomics data across multiple platforms like Biocritis P180, Quant 500, then LCCA, the electrochemistry platform, GCTOF, then LCMS platforms. So that covered lots of small molecules in the serum samples of these patients, serum samples of these patients. And we know that metabolomic levels represent, the impact of diet, lifestyle, social interactions, the genome, the gut microbiome influences, all results in a readout of the metabolome. And metabolome kind of represents the intermediate phenotype that is closest to the clinical phenotype of the patients, right? So we are collecting all this metabolomics data, and that is linked to the neuroimaging data. And we are using metabolomics guided genetic studies to get informed about, genetic variants in these patients. And altogether, we are building pathway models. And then we are doing all these in a system biology approach to see how these are correlated to drug response, why people remit versus why some fail. Then we are also addressing the heterogeneity of the disease, looking at different symptoms of the disease, like anxiety, melancholia, appetite problems with disturbances, etc. And we are also looking at the state and trait markers of the disease. So this one study, which it was a small study, but this showed us for the first time that people who got remission from the disease were not exactly saying or gone back to the healthy control level. So on the left-hand side, you can see here these bar plots. The first bar, box plot, represents the healthy controls. The second one represents the depressed patients. And the third one represents the remitted patients. So patients who had the disease but now are remitted of the disease. So you can see that the remitters were not exactly, nowhere similar to the depressed or the healthy controls. So the study revealed a unique quasi-metabolic state that exists in these people that can be associated with two things, right? Either they represent the cause of the disease, or they can also say something about why some of these people go back or relapse with the disease. So we saw this kind of difference between the remitters and the healthy controls. In several of the metabolites, we studied only the metabolites that were related to the neurotransmitter-related pathways in the brain. Represented by tryptophan and tyrosine and the purine pathways. Now next, we also observed that with the SSRI treatment in these cohorts, there were significant perturbations in the purine pathway, tryptophan pathway, tyrosine pathway, when we treated these patients with these citalopram, antidepressant citalopram or escitalopram. So on the left-hand side, you can see the heat map. So the first column, I don't know whether you can see my pointer, but the left column represents the baseline levels. And we saw the changes from baseline to week 4, and then the changes from week 4 to week 8. So definitely the serotonin levels went down significantly. And then there were other metabolites like the phenolic acid, the tyrosine pathway, then the dopamine metabolite HPG that also was affected, went down in the tyrosine pathway. So the takeaway from this slide that is that with drug exposure, we saw there were significant shifts in the branches of each of these pathways. So, for example, in the tryptophan pathway, we saw that the serotonin level goes down with drug treatment, goes down significantly, but then the indoles, they go up significantly. And this branch is not affected, the kynurenine branch is not affected as much. Similarly, we saw similar trends in the non-canonical branches. They were kind of perturbed more than the canonical branches of the pathways. Why is that? So we tried next to try to correlate some of these changes to changes in the depression severity. And we did not find much association with depression severity, which tells us that these metabolite changes may be due to not only the treatment, but also maybe the patient's recovery or their other diet or lifestyle factors may also be involved. But few of the metabolites showed strong association with change in depression levels, one was serotonin. And when we divided up the samples between responders and non-responders to the drug, we saw that similar metabolites were involved, but there were a few ratios that were significantly associated with the change in depression severity in these patients. So we thought that, okay, we are studying the metabolites or the metabolite ratios one at a time. How about the metabolite interactions? Are these associated with change in depression at all? And this is, we did some graphical network models, and you can see here these highlighted parts, these interactions between metabolites were significantly different between people with high levels of depression versus low levels of depression. And we saw this at baseline, and we also saw this at 8 weeks time. So next, we said, okay, are any of these interactions related associated with any significant genetic variant within these patients? And we found two of these interactions, the methionine tyrosine and also these guanosine and homogentisic acid. These interactions were associated with SNPs. So the strongest association was found with two genes within the locus on chromosome 7. So one was TAP1, which is linked to depression and anxiety, and asparagine synthesis was the other one, which is an important enzyme implicated in neurodevelopmental deficits. So interestingly, patients with this deficiency, they also show modest changes in neurotransmitters. And the signal for the methionine tyrosine interaction was within a locus in chromosome 15 containing the gene AGBL1. And it has a role in controlling the length of the polyglutamate side chain on tubulins. And this process is critical for neuronal survival, and the lack of such control results in neurodegeneration in mice. So these findings underscores the utility of the pharmacometabolomics or metabolomics-informed pharmacogenetic studies to identify candidate genes for further downstream functional analysis. And in fact, Dick Wenschelbaum's group, with our findings, Dick Wenschelbaum's group in Mayo Clinic, they found two significant genes, the ERIC3 and TSPAN5, were associated with the serotonin changes that I showed a few slides back. The serotonin levels goes down significantly with the drug treatment in these depression patients. So they found these two genes were strongly correlated to that serotonin level. And these proteins are highly expressed, found in the central nervous system. And when they knocked out ERIC3 gene in neuronal cell cultures, they found that the serotonin levels went down significantly. And they also found that the ERIC3 genes co-localized with some other proteins, and all of these proteins here, as shown here, were involved in vesicle transport and synaptic vesicle reuptake. So definitely these have implications in neurodegenerative and neuropsychiatric disorders. So it was a nice piece of work that was published in Molecular Psychiatry a few years back. Now, we also interestingly saw in our data that there were significant baseline differences between metabolite levels in these patients. So we did not have control groups. That was one of the limitation of our data sets, both the Mayo and the Emory. So we tried to use the drug as the molecular probe and see how the changes over time happens when the patients are either getting better with the drug treatment or they are not responding to the treatment. So we looked at the metabolite levels at each point in time. And baseline differences are very important because that's when the patients with the drug. So we found this alpha aminoadipic acid, then sarcosin, serotonin, then the short chain acylcarnitine, C3, C5. These all showed significant differences between emitters and the treatment failures. We also found lipids, phospholipids that showed significant changes. We also looked at whether we can see state and trait markers of depression. Now, state markers are something that clearly associates with the disease. When the disease is flared up, these markers also, show up, and when the disease go down, the markers, just are not there anymore. So those are state markers of the disease that tell us about the state of the disease. And the trait markers are something like high cholesterol levels in congestive heart failure. So it may reflect the risk of the disease, but is necessarily associated with the change in the disease symptoms or the disease manifestation. So we're looking for both state and trait markers of depression. So this study is a small study that was published recently. We concentrated on the drug-free arm. Now, the drug treatment actually confounds the metabolite levels considerably. So we saw that the drug-free arm and the remitters and the treatment failures of this drug-free behavioral therapy can shed some light on these state and trait markers of the disease. So now the effect sizes, because this was behavioral therapy, the effect sizes of the changes in the metabolite levels were very small. So what we did was we tried to find, groups of molecules. We know that metabolites, they do not function alone, right? So they function in an interactive fashion. So we found groups of metabolites that were strongly correlated with each other, and we call them modules. And then we from the first the correlation between the metabolites, we did this similar, generating a co-expression network of the metabolites. And then we found closely related, strongly associated modules of the metabolites that were behaving similarly. And then we correlated these strongly related modules to the outcomes. So one of the outcome of interest for our state marker study was whether these modules correlate with the change in depression levels in the patients. And we also found the hub metabolites, or the metabolites that were strongly contributing to each of these modules. So we found 8 different clusters. Each of the clusters are represented in colors. And each of these clusters or modules of metabolites consisted of several metabolites, and these hub metabolites are mentioned here. The purple module was the topmost module, which showed the potential as a state marker panel for the disease, because the metabolites showed strong inverse association with depression levels. And the hub metabolite was C3, or the propion carnitine. So here you can see in this heat map that the purple module, or the cluster, was associated, comprised of carnitine, the short chain acylcarnitines, glutamate, and proline. These were strongly associated with inversely associated with change in depression severity. And two other modules, the yellow module comprised of amino acids, and this green yellow module here that showed a correlation of 0.3 at p-value of 1, comprised mainly of lipids. So if you look at the purple module, we looked at how these metabolites in the purple module are correlated with each other, and also how it related to the HAM-D depression scores. And you can see there were definitely correlations with HAM-D in the first column, and then the metabolites were strongly correlated to each other. If you look at the trajectories of the metabolites, here the panel shows here that HAM-D or depression severity falls drastically in people who remitted of the disease compared to those who did not, the treatment failures. And some of these, all these metabolites show that the remitters drastically decrease over time, as the remitters in blue, they show increase in their metabolite levels in alpha aminoadic acid and all these acylcarnitines. And that showed metabolic resilience present in this patient groups. So we found two other modules that were strongly correlated. Now we are also addressing, I think I'm running out of time, so I will just say that we are also addressing metabolite heterogeneity, and we are finding that metabolites are associated, can explain the metabolic heterogeneity that's present in these patients. And we are finding metabolites that are strongly associated with the different symptoms of the disease. And am I out of time? I cannot hear you properly. Am I running out of time? So I will also show that we are also doing correlating some of our findings with neuroimaging patterns, resting set fMRI images, and we are seeing strong correlations of some of these baseline metabolites, changes in their brain, specific regions in the brain. And I cannot hear any of you. Sorry about that. And we saw several metabolite associations with the neuroimaging patterns in the fMRI. And this is a summary of my presentation. So unique quasi-metabolic state exists in the remitted patients of MDD that is different between the depressed patients and the healthy controls. And radiomics define signatures of treatment exposure and response in MDD. Then our pharmacometabolomics informed pharmacogenetic studies together provided new insights into the response to treatment. And a pilot study with metabolomics provided insights into state versus trait markers of MDD. Metabolomics can define the subphenotypes of MDD and provide mechanistic insights. And gut microbiome, I could not present the gut microbiome related data, but we are also seeing gut microbiome related metabolites are very excited. We are very excited about this prospect that's associated with the symptom severity, various symptoms of MDD. And so that peripheral metabolome inform and complement our neuroimaging measures and may also have value in predicting outcomes to treatment. And I will end with acknowledgements to my students and postdocs who did some of the work and my Duke University collaborators. Emory University collaborators, and Dr. John, he's an eminent psychiatrist, and Helmholtz in Germany, and the entire team of the Mood Disorders Precision Medicine Consortium. This study was funded by the National Institute of Mental Health at NIH. I'm open to any questions. And sorry, I could not hear you. And I hope I did not go too much over time. Thank you.
Thank you, Dr. Bhattacharya, for your delivery of the lecture, which is really informative. There are many questions which has been addressed to you. Among these, I am addressing you 2 questions. Please kindly, if it is possible on your part, to answer. Is COVID-19 a biocidal disease? If answer is no, then why people are afraid about this pandemic situation?
Can you repeat the question, please? I couldn't hear you properly.
Is COVID-19 a biocidal disease? If answer is no, then why people
biocidal disease? What is that?
The question is COVID-19 is biocidal disease? If answer is no, then why people are afraid about this pandemic situation?
There is so much noise, I cannot hear you properly. Sorry.
Okay.
Can somebody please type the question?
How metabolomics?
You can type the question. Yes.
How metabolomics will affect clinical depression of patients in COVID era?
Metabolomics, okay. So, as I said, that metabolomics provides a readout of the impact of, diet, lifestyle factors. So, and medications, and for example, in diabetes patients, you see glucose. Glucose is a metabolite, right? Saying heart disease, see cholesterol, and these are all small molecules, metabolic molecules. So looking at these can definitely, we have to look at them longitudinally over time and associate them with other layers of information. So in our phase 2 of our study, we are trying to put together layers of data from thousands of samples. Our sample size in phase 1 was more exploratory, so sample sizes were limited. So we are now expanding our study to thousands of samples from Europe, and we are going to link the metabolomic findings to transcriptomics and proteomics. So kind of build models of mechanistic models of how this disease is happening. And we're also addressing the heterogeneity in terms of with finite monitoring, we are trying to find these subtypes of the disease which represent more homogeneous clusters of patients. So not each patient have the same trajectory or same manifestation. So we are trying to find these clusters of patients that behave similarly so that we can give them, precise medication and treatments. So I think this is extremely important in this era of COVID-19, which is not going away anytime soon. I don't know, I couldn't hear the question very clearly. I thought this was the question. There is a lot of disturbance in the sound, so I'm sorry about that.
Professor Bhattacharya, there are other questions which will be sending it to you. You can answer them and it will be uploaded in the YouTube of Spark.
Sure, and I just also, before ending, want to mention that if anybody is interested in internships, studies, graduate studies, projects, just email me. I'm happy to respond. And we have lots and lots of data and multimodal data that's available for analysis. So thank you all for this privilege to present. and look forward to hearing from you all.
Thank you, every viewers. Today's session ends here. Tomorrow the session will start at 9:30 sharp. Thank you.
Jetli sir, namaskar. This is Shiv Shankar Das. Jetli sir.